Hi friends, welcome back to my channel. So now we will be discussing about the biology discussion topic which we have seen earlier, active transport. Okay, in active transport we will be seeing it further, types of active transport. Okay, types of active transport. Antipode pumps. We will be seeing antipode pumps in the types of active transport. Okay. Active transport by antipode pumps. Antipode pumps are a type of transmembrane co-transporter protein. So if we see the antipode pumps, they are a type of transmembrane co-transporter protein. Okay, they pump one substance in one direction while transporting another substance in the opposite direction. So if we see the antipode pump, the pump is pumping the substance in one direction and while transporting another substance in opposite direction. Okay, these pumps are extremely efficient because many of them can use one ATP molecule to fuel these two different tasks. So if you see the pump, they are extremely efficient. Okay, many of them are using the ATP molecule, okay, and performing the two different tasks. Okay, one important type of antipode pump is the sodium potassium pump. So example of an antipode pump is a sodium potassium pump, which is discussed in more detail under examples of active transport. So we will be seeing about the sodium potassium pump in more detail in further class under the examples of active transport. Okay, now let's see about the sympod pumps. Sympod pumps take advantage of diffusion gradients to move substances. So if you see the sympod pumps, they take an advantage of a diffusion gradients to move the substances. Okay, to move the substance, they are utilizing the diffusion gradient as an advantage. Okay, diffusion gradients are differences in concentration that cause substances to naturally move from areas of high to low concentration. If we see the diffusion gradients, diffusion gradients are difference in concentration that causes substance to naturally move from higher concentration to lower concentration. Okay. In the case of sympod pump, a substance that wants to move from an area of high concentration to low concentration down its concentration gradient is used to carry another substance against its concentration gradient. In some sympod pump, the substance which want to move from higher concentration to lower concentration, the concentration gradient is used to carry another substance against its concentration gradient. Okay. One example of sympod pump is that of the sodium glucose transport protein. So example of a sympod pump is sodium glucose transport protein. Okay, it's discussed under examples of active transport. So all these examples we will be seeing in further class. Okay, about the sodium glucose transporter, all these comes under the examples of active transport. Okay, active transport by sympod pumps. So you can see here, these are the sympoters. Okay, this is the symptoter, this is also the symptoter, where the two molecules are moving at a time. Okay, endocytosis. In the third type of active transport, large items or large amount of extracellular fluid may be taken into a cell through the process of endocytosis. So in the third type of active transport, large item or large amount of extracellular fluid may be taken into a cell through the process of endocytosis. Okay. In endocytosis, the cell uses protein in its membrane to fold the membrane into the shape of a pocket. So the endocytosis, where the cell is using the protein in its membrane to fold the membrane into the shape of a pocket. Okay, that particular protein is folded into a membrane, folded into a shape where the protein is embedded in the membrane. Okay, this pocket forms around the content to be taken into the cell. So these pockets forms around the content to taken into the cell. Okay. The pocket grows until it is pinched off, reforming the cell membrane around it and trapping the pocket and its contents inside the cell. So the pocket which is growing around the content of the cell, it is going to pinching off from the cell membrane. Okay, so this is the cell where the pocket is forming in the membrane, right? So this pinching off, this membrane is going to pinching off from the cell. Okay, and it is going to separate from the cell membrane around it and trapping the pocket and its content inside the cell. So the contents are filled inside the cell. Okay, these membrane pockets which carry materials inside of or between cells are called vesicles. So the membrane which is pocketed, which is carrying the material inside or between the cells, are called as a vesicle. Okay, we will be calling this as a vesicles. Okay, next, the folding of the cell membrane is accomplished in a mechanism similar to the antipode transport of potassium and sodium ions. So folding of the cell membrane in the mechanism is similar to the antipode transport of potassium and sodium ions. Okay, molecules of ATP binds to protein in the cell membrane, causing them to change their shape. So the molecule of ATP is binding to the protein in the cell membrane and it is causing the change in the shape. The conformational changes of many proteins together change the shape of the cell membrane until a vesicle is created. So the conformational change of the protein is getting changed in the shape of the cell membrane, okay, until the shape is converted into a vesicle form. Okay, in receptive mediated endocytosis, a cell receptor may recognize a specific molecule that the cell wants to take in and form a vesicle around the area where it recognizes the molecule. So if we see the receptor mediated endocytosis, cell receptor which is recognizing a specific molecule, okay, and the cell wants to take it in and form a vesicle around the area where it is going to recognize the molecule. Okay, another type of endocytosis, the cell relies on other cues to recognize and engulf a particular molecule. So in other type of endocytosis, the cell is engulfing a particular molecule. Okay, likewise it is recognizing the cell. Exocytosis. Exocytosis is the opposite of the endocytosis. So in earlier case we have seen endocytosis. Now we will be knowing about the exocytosis. So exocytosis is quite opposite of the endocytosis. Okay, in exocytosis, the cell creates a vesicle to enclose something inside the cell. So if we see the exocytosis, the cell is creating the vesicle to enclose something inside the cell for the purpose of moving it outside of the cell across the membrane. So it is forming inside the cell, and this vesicle is going through the membrane towards the outside, okay, and crossing the membrane to the outside of the cell. This most commonly occurs when a cell wants to export an important product, such as cells that synthesize and export enzymes and hormones that are needed throughout the body. So this is the case where the cell is exporting the important product and the cell is synthesizing the product in the form of enzymes and hormones which is utilized by the body. Okay, so the body needs these enzymes and hormones which is utilized by the body. So the cell is going to release in the form of vesicles. Okay, from inside it is crossing through the membrane and the vesicles are released outside of the cell. Okay, so that particular cell is going to utilized by our body. Okay, in eukaryotic cells, proteins products are made in the endoplasmic reticulum. So in eukaryotic cell, protein products are made in endoplasmic reticulum. Okay, they are often packaged by the endoplasmic reticulum into vesicles and sent to the Golgi apparatus. Now, obviously, proteins are packed in the endoplasmic reticulum in the form of a vesicle, and these vesicles are sent to the Golgi apparatus. Okay, Golgi apparatus can be thought of like a cellular post office. So the Golgi apparatus is working as a post office for our cellular system. It receives packages from the endoplasmic reticulum, processes them and addresses them by adding molecules that will be recognized by receptors on the membrane of the cell intended to receive the product. So this Golgi apparatus is receiving the package from the endoplasmic reticulum and it is processing them, right? And it is going to addressing the molecules to recognized by the receptors on the membrane of the cell to receive the product. Okay. The Golgi apparatus then packages the finished addressed products into vesicles of its own. At last, Golgi apparatus is packing this products into a vesicles. Okay, and these are going to addressed through this Golgi apparatus. Okay, these vesicles move out towards the cell membrane, dock and fuse with it, allowing the vesicle membrane to become part of the cell membrane. So these vesicles are moving towards the cell membrane. They are going to docking with the membrane and fuse with it and allowing the vesicle membrane to become a part of the cell membrane. Okay. The vesicle contents are then spilled into the extracellular space. Then afterwards these vesicles are spilled out into the extracellular space. Okay. So this is the endocytosis where the molecules are enclosed in the membrane. Okay. After that, the molecules are packaged in the form of a vesicles. At last, it is going to pinch off the membrane. Okay. Exocytosis is also same process. Instead, it is going to the vesicle is going to migrate with the membrane, and the membranes are going to through these membranes. The passage of the membranes, the substance are going outside of the membrane. Okay. Endocytosis and exocytosis are the examples of active transport mechanisms. So in the next class we will be seeing about the examples of active transport. Okay. Okay friends, thank you. Thanks for watching. If you like my video, please kindly like, share and subscribe it with your friends also. Thank you so much.